Good day, everyone. So, our topic is about nanoworld and gene therapy. These are the contents that each member will discuss. Number one is discuss the major impacts of nanotechnology on our society, critique the issue on the costs and benefits to society, describe gene therapy in its various forms, assess the issue's potential and detriments to global health, and include the bottom-up versus top-down approach and germline versus somatic gene therapy. So what is nanoworld and gene therapy? It is a platform that includes biology, chemistry, physics, material science, and engineering. Prerequisites for gene therapy include finding the best delivery system, often a virus, typically referred to as a viral vector, for the gene.
Good morning. My name is Kurt Rachel Ramos, and today I will be discussing about nanoscience and the nano world. Before I start, I would like to ask you a question. What do you think is the difference between nanoscience and nanotechnology? Now let's move on. What are nanoscience and nanotechnology? The terms nanoscience and nanotechnology are often used interchangeably, but are two very different things. Nanoscience is the study of structures and materials on an ultra-small scale, and the unique and interesting properties these materials demonstrate. Nanoscience is cross-disciplinary, meaning scientists from a range of fields, including chemistry, physics, biology, medicine, computing, material science, and engineering, are studying it and using it to better understand our world. Nanotechnology, sometimes called molecular manufacturing on the other hand, is the design, production, and application of structures, devices, and systems at the nanoscale. So essentially, one is studying nanomaterials and their properties, and the other is using those materials and properties to create something new or different. Next, let's talk about the nanoscale. In the International System of Units, the prefix nano means 1 billionth or 10 raised to negative 9. Therefore, 1 nanometer is 1 billionth of a meter. For comparison, if one nanoparticle is the same size as a soccer ball, then a soccer ball would be the same size as the Earth. What are nanomaterials? The term nanomaterials has a particular meaning. Nanomaterials are materials at the nanoscale whose properties such as conductivity, color, and mechanical hardness change due to their nanoscale dimensions. Nanomaterials encompass all nanoscale materials, or materials that contain at least one nanoscale structure, either on their surfaces or internally. They can be inorganic, organic, or biological. Nanomaterials such as nanoplates, nanoparticles, nanowires, and nanotubes can be engineered in labs. Nanomaterials can also occur in nature. Naturally occurring nanoparticles include smoke, sea spray, and volcanic ash, as well as minerals, soils, salt particles, and biogenic particles. Nanoparticles, nanowires, nanotubes, and nanoplates are all types of nanomaterials, distinguished by their individual shapes and dimensions. What these materials have in common is that they have one or more dimensions at the nanoscale. Nanoparticles have all three dimensions within the nanoscale. Nanowires and nanotubes have diameters in the nanoscale but can be several hundred nanometers long or even longer. Nanoplates' thickness is at the nanoscale, but their other two dimensions can be quite large. An example of a nanoplate is graphene, a sheet of carbon one atom thick. Nanotechnology has the potential to revolutionize a diverse range of fields, from healthcare to manufacturing. Decades of research and development in nanoscience and nanotechnology have delivered both expected and unexpected benefits for society. Nanotechnology is helping to improve products across a range of areas, including food safety, medicine and healthcare, energy, transportation, communications, environmental protection, and manufacturing. It is being used in the automotive, electronics, and computing industries, and in household products, textiles, cosmetics, the list goes on. Already, there are over 2,000 products on the market that are enhanced with nanotechnology. The ability to tailor the core structures of materials at the nanoscale to achieve specific properties is at the heart of nanotechnology. A few examples of current nanotechnology include the following. Food security. Nanosensors in packaging can detect salmonella and other contaminants in food. Medicine. Some of the most exciting breakthroughs in nanotechnology are occurring in the medical field, allowing medicine to become more personalized, cheaper, safer, and easier to deliver. The potential for nanotechnology to improve drug delivery systems for a range of diseases including cancer, heart disease, diabetes, and other age-related illnesses is an area of intense research for scientists. For example, a 2014 breakthrough saw the development of nanocages, which theoretically deliver cancer-killing drugs directly at the molecular level. This drug delivery method will reduce the dosage amount needed, target cancer cells rather than healthy cells, and reduce side effects. Other exciting developments include the possibility of using nanotechnology to increase the growth of nerve cells, for example, in the damaged brain or spinal cord, and using nanofibers to help regenerate damaged spinal nerves. Energy. Nanotechnology is being used in a range of energy areas to improve the efficiency and cost effectiveness of solar panels, create new kinds of batteries, improve the efficiency of fuel production using better catalysis, and create better lighting systems. Automotive. Nano-engineered materials are in a range of products including high-power chargeable batteries, fuel additives, fuel cells, and improved catalytic converters, which produce cleaner exhaust for longer periods. Environment. Researchers are developing nanostructured filters that can remove virus cells and other impurities from water, which may ultimately help create clean, affordable, and abundant drinking water. A nanofabric paper towel, which can absorb 20 times its weight in oil, can be used for oil spill cleanup operations. Electronics. Many new screen-based appliances, TVs, phones, iPads, and so on, incorporate nanostructured polymer films known as organic light-emitting diodes or OLEDs. These screens are brighter, lighter, and have better picture quality, among other things. Textiles. Nanoscale additives in fabrics help resist staining, wrinkling, and bacteria growth. Cosmetics. Nanoscale materials in a range of cosmetics provide functions such as improved coverage, absorption, and cleansing. In conclusion, nanoscience is an emerging area of science that involves the study of materials on an ultra-small scale and the novel properties that these materials demonstrate. Nanoscience has the potential to reshape the world around us. It could lead to revolutionary breakthroughs in fields ranging from manufacturing to healthcare.
So hi, I'm Principal Lopez. I'll be discussing you today about major impacts of nanotechnology on our society. Nanotechnology. The impact of nanotechnology extends from its medical, ethical, mental, legal, and environmental applications to fields such as engineering, biology, chemistry, computing, materials science, and communication. Nanotechnologies may provide new solutions for the millions of people in developing countries who lack access to basic services such as safe water, reliable energy, healthcare, and education. Military application. A high proportion of nanotechnology research is sponsored by the military and thus focus towards military applications. The potential military applications include nanorobotics, magnetorheological fluid, artificial intelligence, and molecular manufacturing. Intellectual property issues. The patent attorney must establish uniqueness and obviousness in the process of nanotechnology. Patent application. A patent examiner may state that the nanostructured product lacks novelty because the relevant nanostructured material was present in an existing product, even though the nanostructured material was not recognized. Effect on employment. The nanotechnology impact on labor is In its use of particular factors of production during the improvement of nanotechnology, firms are likely to have high demands for the scientists, engineers, and technicians who have to build the integrate the new ideas into processes and products. In addition, there is need for supporting labor services which creates career opportunities. The balance between the citizens and government. The nanotechnology can be expected to concentrate political power in the hands of governments. Nanotechnology can be expected to be applied to further miniaturize and advanced surveillance technologies such as cameras, listening devices, tracking devices, and face and pattern recognition system. So that's it for my report. Thank you.
Take the issue on the cost and the benefits to the society. Content. Here's the two critic issue on the society. Number one, what is the cost on the society? Number two, what is the benefit on the society? The nanotechnology. Nanoscience and nanotechnology are the study and application of extremely small things and can be used across all the science fields, such as chemistry, biology, physics, material science, and engineering. What is the issue cost of the nanotechnology on society? Cost of the nanotechnology. The main problem are the public trust, potential risk, issues of environmental impact, transparency of information, responsible nanoscience and nanotechnologies research. What is the issue benefits of the nanotechnology on the society? Nanotechnology is all about designing, characterization, application, production, and marketing of structure on nanoscale dimensions. The, the interdisciplinary field of nanotechnology is accepted as a window of new opportunities for providing solution, not only in the core technical areas but also promisingly addressing developmental issues like water treatment, eco-recommendation, and sustainable energy. And that's all. And this is the reference.
So, good day everyone. I'm Marian Salcedo and today I will discuss the gene therapy. Here's what you will find in this topic. What is gene therapy? How does gene therapy work? Methods of gene therapy. Discuss the pros and cons of gene therapy. Next. So what is gene therapy? Gene therapy is when a DNA is introduced into a patient to treat a genetic disease. The new DNA contains a functioning gene to correct the effects of a disease-causing mutation. It is also an experimental technique that uses genes to treat or prevent disease. This technique may allow doctors to treat a disorder by inserting a gene into a patient's cells instead of using drugs or surgery. Gene therapy can be a nail for individuals who may have different conditions such as cancer, AIDS, diabetes, heart disease, hemophilia. This treatment strategy is to replace a faulty gene or adds a new gene to nurture or improve your body's ability to fight disease instead of using drugs or surgery. Next. How does gene therapy works? Gene therapy is a therapeutic approach that aims to add, delete, or correct genetic material in order to treat a disease. Modifying genetic material changes how protein or group of proteins is produced by the cell. In other words, gene therapy aims to give the cell a new set of instructions in order to change either the amount of type of protein that is produced. By changing the instructions for the production of protein, gene therapy treats the disease at the genetic level. So doctors may inject the virus containing the faulty gene straight into the affected area of the body. This is useful when only certain populations of cells need to be fixed, for example, cancer disease. Researchers use gene transfer to insert a foreign gene directly into a cancer cells or surrounding tissues. The goal is for the newly inserted gene to either kill cancer cells or prevent cancer cells and surrounding tissue from funneling blood to tumors, depriving them of nutrients necessary for survival. Next. So the two mainly approaches for the transfer of gene in gene therapy is ex vivo gene therapy and in vivo gene therapy. Next. So what is ex vivo gene therapy? Ex vivo gene therapy takes cells from your body, modifies them in a lab, and puts them back into the body. Ex vivo gene therapy involves removing particular cells from the body and sending them to a lab. These cells are changed in the laboratory. A viral vector is frequently used to introduce fresh DNA into the cells. The freshly modified cells are then processed and given back to you, typically by an intravenous infusion. Next. So what is vivo gene therapy? Vivo gene therapy uses viruses or other methods to deliver genes directly into your cells. In vivo gene therapy involves inserting fresh genetic material, typically DNA, directly into your cells. The new genetic material is often introduced into the organism by a vector either directly into the target organ or through an injection into the blood. Next. Here's the pros and cons of gene therapy. The pros of gene therapy is provides options. In case where no other treatments have been effective, gene therapy may be able to cure a patient. Only has to be given one time. Existing gene therapies are given as a one-time dose. This is appealing compared to some other treatments that require multiple doses. Long-lasting effects. Gene therapy has the potential to get rid of a person's symptoms for life. Gene therapy can give many people a better quality of life. Positive effects passed down through generations. Positive effects passed down through generations. If you remove a faulty gene from a parent, they won't transfer this gene to their kids. Rapidly changing technology. Gene therapy is able to exist because of massive advances in technology over the past few decades. Since technology continues to advance, we can expect gene therapy to continue to improve. We can also expect more gene therapies to gain FDA approval. And the cons of gene therapy is expensive. Gene therapy can be extremely pricey, making it inaccessible for some people. Experimental. Gene therapy is relatively new and there's still a lot about it that we don't know. Right now, there are no guarantees. Exactly how successful gene therapy will be is still uncertain. Potentially dangerous. There are some concerns that if gene therapy reach other cells in the body besides the faulty cell, it could have harmful effects. There's also concern that reproductive cells could be changed without detection and this could be passed on people's children. Ethical issues. Some people have these issues with gene therapy. For example, some wonder who will decide what good and bad uses of gene therapy are. Or will gene therapy make us less accepting of people who are different? Lastly is may cause infection. Some gene therapies use inactivated viruses to carry healthy genetic material into a faulty cell in the body. It's possible that these viruses could infect the persons receiving therapy. Next po.
Good morning, my name is Jonathan Gordon and I will be discussing about forms of gene therapy. There are two forms of gene therapy. It's called gene transfer and gene editing. Gene transfer. It is the process wherein doctors transfer a new gene to replace a damaged or inactive gene that causes a problem inside of a cell in our body. In order to do this, doctors inject a carrier or what we call the vectors as the transportation mechanism in order to deliver a modified gene and replace the damaged gene inside the cell. In most cases, vectors are modified viruses. Vectors may also be certain types of bacteria or circular DNA molecules. Now, in this photo, the vector used is an adenovirus. It's a common virus that typically causes mild cold or flu-like illness, but since it's been modified, it's safe to use as a vector to transfer the new gene to replace a damaged one. As you can see in the image, inside the vector is the new gene, and it's been injected inside the body and aims to enter the cell, and then move towards the center of the cell, or what we call the nucleus, where genes are found. After that, the new gene can now serve as a replacement to produce the protein needed to. Another form of gene therapy is called gene editing. It is the process of altering, removing, and replacing genetic materials inside a gene. In this process, what they use are called enzymes. Enzymes are proteins that helps our body in different ways. As you can see in the photo, what they use here are called excision repair enzymes and DNA polymerase. Excision enzymes are responsible for removal of damaged nucleotides. A nucleotide is what we can call as the basic building block of nucleic acids, also known as DNA and RNA. And as we know, genes are made up of either DNA or RNA. What you can see in the image is an example of a gene that is in a straight up figure, not the usual twisted like structure that you know. The strands or lines that are connected is what we call the DNA. And as you can see from the top, there is a damage. That's where the excision repair enzymes come in to remove the damaged strand. After that, it's the DNA polymerase turn to do its job. DNA polymerase are the only enzymes capable of duplicating genetic information stored in the nucleic acid. This is also known as the process of DNA replication. And with that, the gene is fixed and able to produce proteins once again. Thank you for listening.
The bioethics of gene therapy. Good day everyone, my name is Francis Louis Medina. My report is all about the bioethics of gene therapy. Gene therapy in human beings raises a variety of important ethical, legal, and social issues. Somatic gene therapy is similar to other forms of medical treatment, and the goal is to treat, prevent disease in individuals. Raise questions concerning safety and efficacy of treatment and protection for human research subjects. What are the ethical issues surrounding gene therapy? Because gene therapy involves making change to the body basic building blocks which is called DNA. It raises many unique ethical concerns. The ethical questions surrounding gene therapy and genome editing include: number 1, how can good and bad use of gene therapy distinguishes? Number 2, who decide which traits are normal, which is constitute disability or disorder? Number 3, will the high cost of gene therapy make it available only the wealth? Number 4, could the widespread use of gene therapy makes society less accepting of people who are different. Number 5, should people be allowed to use gene therapy to enhance human traits such as high intelligence or athletic ability? Current research on gene therapy treatment has focused on targeting body cells such as bone marrow or blood cells. This type of genetic alternation cannot be passed to a person's children. Gene therapy could be targeted to egg sperm cells, however, which would allow the genetic change to be passed to future generation. This approach is known as germline gene therapy. So the ethical issues associated with germline gene therapy make it unacceptable. Is germline gene therapy ethical? Germline gene therapy targets the reproductive cells, meaning any change made to the DNA will be passed to the next generation. Consequently, the practice has dramatically divided opinion. Do the potential benefits of germline gene therapy outweigh the potential risks? Yes, germline gene therapy enable the correction of disease causing mutation that are certain to be passed on from generation to generation, sparing future generation from suffering from the disease. No, the effects of gene therapy are too unpredictable. Even if the therapy successfully cures the disease, other mutation can potentially be introduced. Those germline gene therapy considered an individual's humans, right? Yes. No unborn child, whether conceived naturally or artificially through in germline gene therapy is able to choose their genetics and whenever they are born with or without particular condition. No, individuals produced through germline gene therapy cannot give their consent for genetic material to modify. Is germline gene therapy ethically acceptable? Yes, gene therapy advisory committee advice on the ethical acceptable proposal for gene therapy research on humans. No, this widespread use of gene therapy make me society less accepting of people who are different or who have a particular disability or genetic condition. Is germline gene therapy affordable? Yes, if germline gene therapy has the potential to remove a disease completely from the population, it will remove the long-term healthcare costs of treating the disease. No, in areas of the world without a free health service, only parents who can afford the suitable health insurance will be able to undergo germline gene therapy. Poor parents will not have access to this form of therapy. Is germline gene therapy really needed? Yes. With the widespread use of germline gene therapy, particular genetic condition could be eliminated from population altogether. Condition like Huntington disease could become disease of the past. In the same way, the global vaccination program have eradicated disease like polio. No, germline gene therapy would never be effective without the role of the need for the post-fertilization screening of embryos. The therapy enhancement distinction is not as clear as some assume because the concept of disease is not properly objective and includes social, cultural, and ethical dimension. Investigator and institution should take appropriate steps to protect the rights welfare of gene therapy. Patient participating in clinical research. In thinking about the balance of benefit and risk of gene therapy study, it is important to consider the safety and efficacy of the proposed research and ability of alternatives.
Good day everyone, I'm Macraven Guerrero and I will discussing about the risks and benefits of nanotechnology. There is a lot of discussion about nanotechnology as enormous technology and the economical potential of the world. Also, there are concerns about the new risks that nanotechnology could bring for human and environment. People are concerned that nanotechnology may increase the gap between developed and developing countries. So far, it seems that most nanotechnology applications do have Western bias. For example, sunscreen and sports equipment, and computers. However, it is not inevitable that this trend will continue. Applied nanotechnology could benefit all, particularly in the areas of medicine, energy resources, and information communication technology. Under nanomedicine advance in this area could obviously benefit all as long as they are available to advance such as biocompatible nanostructured surface on replacement body parts, for example, hip, hip joints, or heart valves, make them accepted by the body. Targeted drugs, nano-sized capsules which go to straight to the infected area of the body, reducing unwanted side effects. Nanostructure materials for bones, tissue, and even nerve regeneration. Nanosized sensors, which can analyze our blood for viruses and give back results almost immediately, so-called a lab on a chip. This tiny device could be especially useful in developing countries to save on time and personnel. There are concerns that this kind of medical advance may only be available to developed countries or to the people that can afford it. However, nanotechnologies often have the advantage of being relatively cheap to produce once they have been designed. For example, a nanotechnology-based tuberculosis diagnostic kit is being developed in India, which is highly efficient and compact, but will eventually only cost 30 rupees, which is less than $1. Medical breakthrough always exciting, but also bring risks that need to be searched and managed. For example, some nanomedical devices are 100 to 10,000 times smaller than human cells. Therefore, we need to know how they have within the body before we can use them safely. Of course, it's being small that gives them unique potential to deliver targeted treatment, but we need to manage this potential. Let's now discuss about energy and resources, in which climate change and diminishing resources are global concern. Nanotechnology is often cited as possibly having the solution to some of the world's biggest problems. Solar cell, which is smaller, yes, more efficient and cheap to manufacture, could make solar power a credible alternative energy source, like a nanocrystal bulb that uses 100% of this energy within nothing waste in heat. Carbon nanotubes, 10 times stronger, that still a sixth of weight. These tiny tubes could revolutionize the materials we use and energy we consume. Water. Nanoparticles cost to remove toxin from water to make it drinkable and save many lives. Finding answer to energy problems is something that hopefully will benefit everyone. However, there is still concern that some tailored to Western needs solution which work for an already developed society with an infrastructure in place. Another issue is that of manufacturing. Developing countries want to ensure that they become producers of nanotechnology as well, not solely importing the technology from developed countries. Anyways, anyways needs to be managed correctly to avoid polluting our world. Waste from nanotechnology to be managed also accept that some parts are so small that they cannot be dealt with by con- conventional methods. New ways of disposal needs to devise so our environment is not threatened and nanotechnology can develop safely. Let us proceed to information communication. Nanotechnology could one day use single electrons to work processors. This could make computers 100 times faster with huge memories and many applications. Nano-sized ID tags could in the future be attached to everything we buy, giving information about what were consumed. In theory, this could cut down in waste and theft, but it could also raise concern about individuals' property. Information and communication technology. Nanotechnology is creating smaller microprocessors. This means the devices are more compact, more portable, faster, more powerful, and contain many more applications. Nanotechnology is giving scientists the ultimate opportunity to optimally design and create new materials. With these new opportunities come responsibilities, and it's up to all of us to use them safely, yet still allow technology to develop and be useful. We have responsibility too. By keeping informed and engaged about the progression of nanotechnologies, we can make educated contributions to future decision making regarding the use of nanotechnology. That's all for my report. Thank you for listening.
So, good morning everyone. I am Prince Mark P. Castañeda and the reporter for the bottom-up versus top-down approach. So, let's go. So, the content of my topic is what is the bottom-up approach? Number 2, what is the top-down approach? And number 3, differences between bottom-up and top-down approach. And for the last one is the examples of bottom-up and top-down approach. So, what is bottom-up approach? Bottom-up analysis is an entirely different approach. In general, the bottom-up approach focuses on specific characteristics and micro-attributes of a single stock. Many of these techniques are still in development or are only now being used commercially to produce nanopowders. So, the bottom-up approach refers to the construction of material from the ground to up, atom by atom, molecule by molecule, or cluster by cluster. So the bottom-up approach is the alternative approach which has the potential of creating less waste and hence the more economical or more freely to use. So this was the representation of bottom-up approach. So at first these are the atoms and after that they will become cluster and after that they will become nanoparticles. So it was small to large. So, for the top-down approach, what is top-down approach? Top-down analysis is the use of comprehensive factors as a basis for decision making. The top-down approach seeks to identify the big picture as well as all of its constituent parts. These elements are typically the driving force behind the end goal. The top-down approach is to miniaturizing existing technologies. So, the top-down approach involves the breaking down of the bulk material into nano-sized structures or particles. So it was like a big bulk material and then they will break it into small pieces. So this was the representation of the top-down approach. Here is the bulk metals, then they will become powder and they will become nanoparticles. So let's go to the differences between the two or the bottom-up approach versus top-down approach. There are two distinct paths to take. The first is the top-down approach that involves miniaturizing existing technologies or they will belittle the technology. While the second is a bottom-up approach that involves building increasingly complex molecular device atom by atom or they will build up to become a big. In the long run, the combination of top-down and bottom-up techniques is expected to provide the best combinations of tools for nanofabrication. The development of new fabrication and measurement tools is required by nanotechnology. So, bottom-up approach are best suited for assembly and establishing short-range order at nanoscale dimensions. Whereas top-down approach are best suited for producing structures with long-range order and making macroscopic connections. So let's go to the examples of bottom-up and top-down approach. So the first one is the organometallic chemical route. So organometallic is a molecule that contains a metal atom bonded to a carbon atom compounds with, for example, metal nitrogen, metal oxygen, metal phosphorus. Bonds are defined as coordination complexes but are often described as organometallic. So the organometallic chemical route is like a combination of carbon atoms. Next. So the next example of bottom up approach is sol gel synthesis. So the sol gel synthesis is process is more chemical method for producing various nanostructures, particularly metal oxide nanoparticles. In this method, the molecular precursor, usually the metal alkoxide, is dissolved in water or alcohol and then heated and stirred to form a gel via hydrolysis or alcoholysis. So this was the representation of it. So let's go to the top-down approach example. The first top-down approach example is the ball mill. Ball mills are commonly used to crush and grind materials into extremely fine powders. A hollow cylindrical shell rotates about its axis in the ball mill. The cylinder is filled with balls made of stainless steel or rubber depending on the material contained within it. So this example is like you are crushing a big particles and turn it into small particles. So for the last example of the top-down approach is the liquid phase exfoliation. The liquid phase exfoliation or the LPE disperse graphite flakes into a solution using intercalating agents, which are molecules that place themselves between layers and surfactants. Mechanical processing steps such as controlled ball milling, as well as solvothermal or sonochemical processes help to support it. So, lastly, for the conclusion of this topic, the biggest problem with the top-down approach is the imperfection of the surface structure. Such imperfections will have a significant impact on the physical and chemical properties of nanostructures and nanomaterials. Among the two, or the bottom-up and the top-down approach, the bottom-up approach is more accepted in synthesis of nanoparticles due to many merits as fewer defects, more homogeneous chemical composition, and better ordering. And that's all for my topic. Thank you.
So hello, our last topic is about germline versus somatic gene therapy. So here's what you'll find in this topic. What is germline gene therapy? What is somatic gene therapy? And germline versus somatic gene therapy. So hello, good morning. I'm Angelica T. Mangrobang, your last presenter. Do you think that gene therapies are good or bad? So let's go to first one. What is germline? So germline gene therapy is when DNA is transferred into the cells that produce reproductive cells, eggs, or sperm in the body. This type of therapy allows for the correction of disease-causing gene variants that are certain to be passed down from generation to generation. So germline gene therapy is the correction of diseases that we can inherit in the future. It fights the hereditary disorders occurs at humans are possibly inherited from the germline cells. So next is, what is the process of germline gene therapy? A new gene is inserted directly into a cell. A carrier called a vector is genetically engineered to deliver the gene. An adenovirus introduces the DNA into the nucleus of the cell, but the DNA is not integrated into a chromosomes. So germline targets reproductive cells in our body. To emphasize, germline gene therapy only utilizes reproductive cells, egg cells, and sperm cells. It is inserted the desirable DNA into the cells in a laboratory setting and then return the modified DNA or genes to the patient. Next is, what is somatic? So somatic gene therapy can be defined as the ability to introduce genetic material, RNA, into an appropriate cell type or tissues in vivo, in such a way that it alters the cell's pattern of gene expression to produce the therapeutic effect. So somatic gene therapy pinpoints on the improvement of genetic disease by handling of non-reproductive or somatic tissues. This method of gene therapy includes the exclusion of some of the dysfunctional cells and introducing them with a cloned wild-type gene. This type of gene therapies cannot pass through generations because it only helps the non-reproductive system. So next is the process of somatic gene therapy. So gene therapy methods can be used to pursue different goals. The introduction of so-called transgenes into tissues or cells can be aimed directly at correcting or replacing defective or missing gene functions. This primarily treats monogenic hereditary disease. So, however, there are some major technical obstacles that must be overcome before somatic gene therapy is likely to work. The first is acquiring the human gene by the patient's cells. So you should check. So checking the gene if it is compatible, or I'll take some more findings about his cells or genes. The second is hurdle is insertion of gene into the right cells. So wrong insertion of gene can lead to a bad conditions that I will tackle later. Then the final obstacle is making sure that the gene is active. So this means it must be under the control of sequence of DNA that is responsible for switching the gene on. Next is, let's go to germline versus somatic. So germline, the cells that form eggs in females and sperm in males. And somatic is any biological cell forming the body of multicellular organisms. Now let's compare germline and somatic. So let's talk about the risk of getting a gene therapy. So let's start with germline. So germline gene therapy are too unpredictable. Even if the therapy successfully cures the disease, other mutation can potentially be introduced because germline gene therapy targets the reproductive cells. Any additional mutation that are introduced as well as result will be passed on to the next generation. So in germline gene therapy, we still can't be sure that the outcome will be good because there may be a new mutation in your cells that we don't know until it passes the offspring or child. It may be better or worse than what you treated before. So there's an instance when it leads like that. So the cell may not be well taken care of, or the medicine is not taken consecutively, or it can be also how your body reacts on that new cells. So let's go to somatic. So somatic gene therapy, it's possible that once introduced into the body, the viruses may recover their original ability to cause disease, possibly of causing a tumor. If the new gene get inserted in the wrong spot in your DNA, there is a chance that the insertion may lead to tumor formation. So somatic is more carefully done because if there is a mistake in the insertion, it may lead to a tumor. And we are also not sure because the virus or the diseases may still return on its own. Maybe the body couldn't handle it, or lack of findings about his or her gene, so it didn't react well and went back to the way it was before. So the most riskier gene therapy is somatic. So next is, let's compare the effectiveness of those gene therapies. So the first one is, germline gene therapy is contained only in the target cell type. No other type of cells are affected. It can be more effective and can be a permanent cure for genetic diseases that run in the family. It has the potential to eliminate a disease from the population. So, germline is effective because the target is the cell that can be passed on to the next generation. They target the reproductive cells only, so there is not much concern about the outcome. It is the most used gene therapy because it cures hereditary diseases. So next is somatic gene therapy, targets body tissues. These cells do not produce sperm or eggs. Therefore, the gene is not transferred to the next generation. So in somatic gene therapy, it is effective also, but the target only is the tissues of the body. It cannot pass to other generations because it treats only dysfunctional parts of the body. So the most effective is germline gene therapy. So next, so our last, the cost, which is the most expensive. So for germline, the average of gene therapy is likely to cost at least 1.5 million. Oh, that's a lot. So next, in somatic gene therapy, currently the cost of human genome editing therapies range between $373,000 and $2.1 million. So as you can see, that is just an estimated cost. It depends on the diseases you want to cure or the effectiveness that you like. So in this estimation, somatic is the most expensive because it is the most riskier to do and it targets all the non-reproductive system or body tissues. So the least is germline because it only targets cells, the productive cells in our body. So in short, germline gene therapy treats the cells in our body, which is the reproductive cells, the sperm and egg cells. And somatic gene therapy treats body tissues and dysfunctional diseases. And that's all for germline and somatic gene therapy. As a student about these issues in science, technology, and society, in terms of nanoworld and gene therapy, I want you to encourage everyone to be aware about the positive and negative effects. It's so good that we're having a new technologies to treat our diseases. Those we don't want to inherit in the future, it can be replaced or repaired. But we can ensure about our safety or the outcome that we want. It can also make a new mutations that is not good for us or for our health. And in the end, we will regret it. In the future, science, technology, and society is a big help in our lives. But we should use it in a good way so that we can abuse the power of technology that it has.
So, what can we do to mitigate and control the use of science and technology? In recent decades, scientific research has produced unexpected new knowledge and technologies that provide unprecedented opportunities to improve human and animal health and environmental condition. However, some science and technology can be used for both destructive and constructive purposes when it comes to dual use. And the misuse of science and technology, scientists bear a special responsibility. Yes, technological advances help us with our daily life, with our daily living every day. They can do anything that they want without being tired. So to control or mitigate the use of science and technology to prevent bad outcome, we will reduce by reusing, recycling, and using fewer resources like oceans, fresh water, and air, all shared resources that must be protected through careful planning and regulation of technological systems. One example to control or mitigate the usage of science and technology when you are capturing a sunset, sunrise, or your friends bonding. You don't need to take a lot of pictures of it. You can just mesmerize the beauty of the sun every day. And you can keep all the memories and enjoyment by yourself instead capturing it. Or just take one picture that you and your friends are all happy. So to to do this, you must have a awareness, safety and security, education and information. And that's all. Thank you.
Importance of science and technology in our society. It made our life easier and simple because it helped in reducing our time and effort. For example, the transportation process we use. We can reach our destination quickly, and the mobile devices like cell phones, we can communicate or talk to our loved ones even if they are far. In science, it helped our society to fulfill the human needs, and because of science, our life will become longer and healthier. For examples, the medicines, it helps us to cure our disease, which it can make our life strong. Without science, we wouldn't have technologies like cell phones, computers, cars, etc. Because of science and technology, it changed the way we live, and humans now have to live in a simple life.
Information technology, including the internet, is an excellent gift of technology. With the help of the internet, not only do we get tremendous knowledge in science and other materials, but we can also keep communicating with our friends and families constantly. The internet and related tools are already playing major and important roles in areas like data mapping and visualization, crowdsourcing, and more. Technology is also important in business because transactions and other events take place through the computer.